This is the third recording for chapter 1. So we are starting on slide number 39. And we are actually going to move. This is just information about interactions between organisms with their physical environment and with each other. Okay. And talks a little bit about species that are threatened by global warming, but I'm going to move on to, this is slide number 44, and talk just a little bit, very briefly, about evolution. So the definition for evolution sorry, it didn't give us a good definition, so I'll just have to tell you. Okay, so here it is. I'm so sorry. Okay, so evolution is the concept that living organisms are modified descendants of common ancestors. It also can be defined as the change in the characteristics of species over time. So changing characteristics of species over time is evolution, and we are going to go on and talk a little bit about the diversity of living things. So there are 3 domains of living things. There are 3 domains. So all living things are grouped into one of these 3 domains: bacteria, archaea, and eukarya. So you definitely will need to know these 3 domains, be familiar with them, not just for Bio 111, but also for Bio 112. The organisms that are members of bacteria and archaea are prokaryotes. And that means that they are unicellular and their cells don't have a nucleus. And then the domain eukarya consists only of eukaryotic organisms. Some of them are unicellular, but mostly they're multicellular. So there's 4 subgroups or kingdoms of living things within the domain eukarya. That would be the plants, which are always multicellular and they carry out photosynthesis. Fungi, which are almost always multicellular. There are unicellular fungi, and those are the yeast. Yeast are unicellular fungi, but the rest of the fungi are multicellular, and they obtain their food by absorption. Animals ingest their food, and they're always multicellular. And then the kingdom Protista includes protozoans, which are unicellular, and algae, which can be unicellular or multicellular. So we have the kingdom Plantae, which is always multicellular organisms that carry out photosynthesis. Fungi, which are almost all multicellular, and they absorb their food. Animals, which are always multicellular, and they get their food by ingestion. And the kingdom Protista, which can be unicellular or multicellular. The unicellular protozoans, and then the algae can be unicellular or multicellular. Okay, so the kingdom Protista, it mentions here that they are mostly single-celled or unicellular. They're the most numerous and diverse of the eukaryotes. Basically, the kingdom Protista is either protozoans or algae. Protozoans are like the little creatures that you see in pond water under the microscope. So here's a picture. This shows you some protozoans. So we have within all the 3 domains are shown in this picture. So we have domain bacteria, domain archaea. All of these 2, for these 2 domains, they're always going to be unicellular, and the cells are prokaryotic. meaning that they don't have a nucleus. And then for the third domain, the eukarya, we have 4 kingdoms: the plantae, the fungi, the animalia, and the protist. So eukarya is always going to have cells that have a nucleus. Some are multicellular, some are unicellular. So I'm skipping some of this, and then we're going to just briefly talk about Charles Darwin and natural selection. Charles Darwin published a book called On the Origin of Species by Means of Natural Selection, and he published that book in 1859. And he made 2 points. He said that the species that he studied on the Galapagos Islands and in South America showed evidence of descent with modification. In other words, they descended from a common ancestor, but they were a little bit different from that ancestor. So that's what descent with modification means. So that was his first point, is that he observed animals and plants on the Galapagos that showed evidence of descent with modification from the common ancestor that would be found on the mainland of South America. And then his second point was that the way that they got those modifications is through natural selection. Natural selection is the cause for descent with modification. So, this is a picture of the title page of his book. And there's a little bit of information about Darwin and the finches that he studied. I'll go back to that picture. Well, no, it just shows that there's several different species of birds that are shown here. So just to make the point that birds originated from a common ancestor, but they're not all alike. So they have, all birds have modifications from the common bird ancestor. For example, penguins can't fly. You know, the flamingos are pink, and they have long, kind of flexible necks. So we have some birds of prey, and then raptors, and then we have birds that eat seeds. So we have lots of different variety in birds. And this is a picture. This is slide number 60, and it shows how natural selection works. So if you look at the first picture, picture number 1 shows you that you have these little beetles and that they have variation in their traits. Some of them are very light colored, some are medium colored, and some are dark. So most likely the ones that get eaten by this bird that eats insects, they're going to be the ones the bird can see. So they're going to be the lighter colored ones since the background is dark. Okay, so you can see the bird eating a lighter colored beetle. So the traits that are most suited for survival are going to be the ones that tend to get passed on to the offspring. So if the bird eats mostly the light colored beetles, then what's left is mostly dark colored. And over time, year after year after year, when you have new populations of beetles, you're going to start seeing more of the dark colored beetles. And that is how natural selection works. Adaptations are structures in an animal in an organism that allow it to have like a survival benefit. So for example, the wings on this bat are adaptations to allow it to fly. So an adaptation is just defined as something that gives the organism a survival benefit. Let's see. There's a note here on the finches that Darwin studied in the Galapagos, that the finches they're descended from a common ancestor, but all of the ones that live on the different Galapagos Islands have different beak shapes. Here we go. Here's a picture. I was looking for this. So this would be an illustration of the ancestral finch to all these different varieties of finches. Some of them have these big, thick, heavy beaks, and those are going to eat like nuts and seeds. Some have long, slender beaks, and they're going to eat insects. And then some of them have beaks for eating smaller seeds. So the beaks are designed for the food sources on the habitat where they live. Okay, so now we're going to talk about the scientific method. The scientific method is used to design experiments and to solve problems, to answer questions in science. Sometimes we look at qualitative data and sometimes we look at quantitative data. Sometimes we look at a mixture, but data, if we call it quantitative, it's just numerical. They're numbers that were measured, and we can organize quantitative data into tables and graphs, but qualitative data is more like descriptions most of the time. Okay, so Jane Goodall here, she's very famous. She studied chimpanzees, very famous for studying chimpanzee behavior. And she is observing right now and just writing down what she observed. So she's writing down descriptions of what she observed. So this is qualitative data, observing and recording descriptions of the chimpanzee behavior. But quantitative data would be like if she was taking measurements somehow. Maybe she was measuring the weight, she could be measuring their weight. She could be doing blood work, but anything that involved numbers would be quantitative data. And then in science, we also use two different types of reasoning. There's inductive and deductive. So inductive reasoning is used to derive generalizations from a large number of observations. So to make a generalization from a large number of observations. So that's inductive reasoning. And when we formulate a hypothesis, we are using inductive reasoning. Okay, so a hypothesis is an explanation based on observations and assumptions that leads to a testable prediction. So hypothesis is an explanation for whatever it is that you're observing. And it may be a true explanation, or it may not be, but that's why we test it. So what we can do with a hypothesis, by the way, a hypothesis is always a statement, but we can design an experiment to test our hypothesis. And once we design our experiment, we can make a prediction on the results of that experiment. So here's an example. If the desk lamp doesn't work, your question is, why doesn't it work? You can formulate hypotheses. You can say hypothesis 1, the bulb is burnt out. Hypothesis 2, the bulb is not screwed in properly. Both of these are statements. Both of these can be tested, right? So they're explanations for why the lamp doesn't work. So that's what a hypothesis is. It's a statement that explains the question that you have, and it's something you can design a test, to determine if it is true or not. So if we look at the scientific process, continuing on with this example, if we test hypothesis number 1, if the bulb is burnt out, then our prediction is replacing the bulb will fix the problem. If we replace the bulb and the lamp works, that supports our hypothesis. But if we replace the bulb and it doesn't work, then it does not support our hypothesis. So always make sure when you record your results as to the results of your experiment, you're always going to say either the results supported the hypothesis or the results do not support the hypothesis. You're not going to make statements like the hypothesis was proven true, or the hypothesis was false, you're just going to say whether or not your results support it or do not support it. Okay? Now, inductive reasoning is the kind of reasoning we use when we make a hypothesis. But when we form a prediction, that is deductive reasoning. Deductive reasoning uses generalizations or general premises to make specific predictions. So when you make a hypothesis, you're using inductive, and when you make a prediction, you're using deductive. All right, and we've already said the hypothesis has to be a statement, so it has to end with a period, not a question mark, and it must be testable. You must be able to design some kind of a test that will support or not support the hypothesis. The scientific method is the problem-solving method that we use in science to answer questions. All right, here is an example of an experiment that was actually done using two different species of mice. You can see one is brown and one's white. Well, it's really like sandy colored. The light colored one lives on the beach where the sand is real light colored. And then the dark colored mouse lives inland where the land is darker. Okay, so the prediction was the hypothesis was, not the prediction, the hypothesis was that the color patterns in the mice had evolved as adaptations to protect the mice from predators. And that makes sense, and that's probably is true, but how do we prove that? So that's the hypothesis, is that their color patterns have evolved as adaptations to protect them from predators. So the test, the researchers basically made a prediction that if you put mice of different colors in the wrong habitat, that they would get eaten more often. So that's what they did. They basically, they built models of mice instead of using live mice, so that was kind of them. They built models of mice that were painted to match the surroundings, and they placed equal numbers of each type of model in each habitat, and they recorded signs of the mice being attacked, like, teeth marks, that kind of thing. So the results showed that the camouflage does appear to provide them with protection from predators. So in the beach habitat, the dark models were mostly attacked. The light models were not, because they're harder to see, as you can see here. And then in the inland habitat, the light models were mostly attacked. So that supported the hypothesis that the coloration of the rock pocket mouse is designed to protect it from predators. Now, this is a video, slide number 83. It's a video that you can watch. It's a good video, but I can't show it in the recording. I also want to go over with you real quick, when you do design a controlled experiment, you got to have a control and you got to have an experimental group. So the control group is going to be in the mice experiment, the control group is going to be the ones that their coloration matches the habitat. So the camouflaged mice are the control group, and the experimental group are the ones that their color doesn't match the habitat. They're not camouflaged. So you have an experimental group and a control group. Sometimes you can have more than one experimental group, but in this experiment, we had one experimental group, one control group. So the control group is always the camouflaged mice. And anytime you design a controlled experiment, the control is going to be what you compare your experimental group to. Okay, so it's used for comparison. All right, then we have the independent and the dependent variable. So you need to make sure that you can read an experiment and tell me which is the independent variable, which is the dependent. So dependent to me is easier. The dependent variable is the one that you measure. It says here it's the one that's predicted to be affected in the response, but I always think of it as this is the variable we are measuring. So in this case, we were measuring the predation, like the attacks on the mice. So the amount of predation was the dependent variable. That's what we were measuring. Independent is the one that you are testing, that you are manipulating. As the researcher, it's the variable that you are actually manipulating. So what did the researchers do? They placed either camouflaged mice or non-camouflaged mice. They placed equal amounts of both into the different habitats. So the independent variable would be the coloration of the mice. That's the part that the researcher is manipulating. Dependent variable is always what you are measuring. Okay, make sure that you know the definition of a theory. A scientific theory is supported by a large body of evidence, and it's generally thought of to be true. We haven't got any evidence that it isn't true, and lots of evidence that it is true. So a scientific theory is also an explanation to a question, but it's got a lot more support than a hypothesis does. Okay. And there is a section on science and technology, and that is it for chapter 1.